Hello, everyone. My name is Favour Ebiala. I worked in the Kalume lab this summer. I'll be presenting on the impact of MITO 109 on swallow breathing, discoordination in a mouse model, and Leigh syndrome. So what exactly is Leigh syndrome? Leigh syndrome is a mitochondrial disease, specifically a neurodegenerative disorder with a severe CNS affectation. In an MRI scan, as you can see in the photograph, it presents as a bilateral symmetrical lesion within the basal ganglia and the brainstem. In terms of symptoms, it typically presents as motor and respiratory deficits, usually accompanied with seizures and sudden death. Specifically in Leigh syndrome, we will be focusing on the endospore gene, which is a gene located in the complex 1 of the mitochondrial electron transport chain. Mutation in this particular area is associated with early onset Leigh syndrome. Unfortunately, there's no current treatment for Leigh syndrome, so we're hoping that our research would aid in that. With the endospore knockout mutations, they typically differ in their expressions, usually based off of the neuron types. With the GABAergic neurons, they usually express as seizure activity. With the glutamatergic neurons, which is what we'll be focusing on, they express as motor and respiratory impairments. I do want to point out that there are different types of knockouts. For this particular project, we focused on cell-specific knockouts in the glutamatergic neurons in the brainstem. So with our colleagues at MITO Therapeutics, they are testing out a new drug. It's been showing promising results in the preclinical studies using the global model. So we hypothesized that the MITO 109 will rescue swallow breathing discoordination and prolong life in our LS. So here's just an illustration of how we generate our mouse model. Specifically, we'll be using the homozygous endospore knockout BGLU2 Cre positive. As you can see on the right side, we have the treatment and the no treatment group. These mice have the exact same genotype. The only difference between the two is that we will be using the treatment on one and the other is just a control. So for my methods, with the drug, we use the MITO 109 drug, and we use peanut butter as our drug vehicle. For the no treatment group, we only use peanut butter, and we administered 0.2 cc to each mice. The way we administered the peanut butter was by applying it to the side of the cage. With the treatment group, we administered peanut butter and MITO 109 mixed together at a dosage of 220 mg per kg. We started dosage at P21, which is just day 21, which is the day that the mice are weaned. The drug was administered once every day during the weekdays only. We didn't administer it during the weekends based off of the company recommendations. So for our results, as you can see on the left side, with the no treatment group, you can see a steady increase in their body weight and then kind of a decrease starting at week 10. I do want to point out specifically K900, which is the orange on the left side. That particular mouse died before week 12 under anesthesia. And we noticed that the mouse, its body weight decreased a little more than the rest. And we suspect that the anesthesia was an overdose, that the mouse overdosed on anesthesia based off of the low body weight. With the other mice, with K1215 and K1274, those mice are much younger. They haven't been experimented on just yet, but they're doing well. So if you move on to the right side on the treatment groups, you can see that there's one mice that lived longer than the rest, and that was K865. And that was our oldest mice out of both groups. That mice lived about 113 days, which was a good sign. And then with the other two, the shorter ones, the K1273 and K1292, those are also very young, but they're also doing well. For our calculated p-values, the body weight was 0.06, and for the lifespan, it was 0.07. So based off of what we saw on the previous slide, you can see that the weight decrease started about week 10 or 11, and that's usually the time they got sicker. And then week 12 is when we started with our experimentation. But overall, the treatment group did live longer than the control group, and our data wasn't necessarily statistically significant because it's still an ongoing study, but we do hope that it could lead to possible treatment for Leigh syndrome in human beings. I just want to give a special thank you to Dr. Kalume, Dr. Alyssa Huff. I also wanted to thank Dr. Ellenbogen, Mrs. Ellenbogen, Sylvia, and Julie, and everyone else that made the program. Thank you.